help us detect the presence of the lipid droplets. So from the data, this is the untreated cells where we have the presence of the kinase indicated in red, and then stained with BODP where we have the lipid droplets indicated in green, and upon co-localization you see the presence of the kinase on the droplet. However, you see that they come back to the cells. So this kind of gives us the idea that in addition to the loss of the kinase, there's also the loss to them. They were now treated with BODP, and then the fluorescence intensity from the BODP was then measured using the flow cytometer. So here, this graph here shows the fluorescent intensity that was measured. And for the purple chart here, we have the untreated and unstained, where there is no or less signal. And upon treatment, upon staining with the BODP in a normal cell or untreated cell, we see a shift or an increase in signal intensity, indicating the presence of these neutral lipids, as well as, which is suggestive of the presence of lipid droplets. However, upon treatment with the gallets, we observe a reduction in the fluorescence intensity that is suggestive of a reduction in this neutral lipids, as well as a reduction in the lipid droplets. But this process was then recovered slowly during a 2-hour recovery. So this buttress the point that these alkyl gallets also cause a disruption or loss to the lipid droplets in addition to the lipid droplet kinase. So with this data in mind, we are able to show that alkyl gallets, when added to the cells, are able to cause a disruption to the kinase as well as the droplets. So we had two models that could help us explain how these compounds could be interacting to obtain this phenotype. So the first model is a block in the lipid droplet synthesis. So here we hypothesize that these gallets could be interacting with key metabolic pathways, and that could prevent the formation of molecules that are needed to form this lipid drops. The idea is that perhaps these compounds could also be interfering with downstream effects, causing kind of a feedback inhibitory loop that will force the cells to use up its lipid droplet content, causing a release of this triacylglyceride as well as the sterol esters to create precursors needed to buttress the locked pathway, and that could lead to the disruption of the lipid droplets and then the subsequent phenotype that we see. So in conclusion, alkyl gallets, we're able to show that alkyl gallets cause the loss or disruption to the lipid droplets as well as its kinase. And more importantly, this data tells us that these compounds could be used to further study lipid metabolism in trypanosomes. So I'd like to thank and acknowledge the following people or the funding agencies and institutions that have supported me to bring this work this far. And I'd also like to thank you for your attention.
Okay. We have time for 2 questions.
Very nice talk, Cynthia.
Thank you.
How conserved is this kinase with other trypanosomatids? And does your alkyl gallet work in those cells?
So as at now, we don't know. Okay, to the best of my knowledge, I don't know how the alkyl gallet works in the other cells. So this kind of a new information that is coming up. And for the conservation, they're also found in other the other kinase like Leishmania and T. cruzi. And yeah.
Hello, Cynthia.
Hello.
Can I ask, when the lipid went down within half an hour from BODP staining, do you think that represented a loss of neutral lipids from the cell, or is it a product of the staining? So the lipids are the same, it's just the staining is less because they're not in foci.
I think I'll go towards the loss of the lipid content because, I mean, we included controls. So we had the untreated, we had also a compound that is not really a gallate, and we showed that this phenotype is not observed. So I will go towards the effect of the compound other than the staining with the BODP. Yeah.
We have time for a few more questions, maybe one or two more. Okay, thank you very much, Cynthia.
So it appears our sponsors all want to present tomorrow and Wednesday, so that means that we're a little bit ahead of time. And I'd like to thank the chairs for this session, Dr. Akins and Dr. Aniwe. And we'll move right on to the next session, which is session 4, Drug Discovery Mechanisms and Resistance, section 1. And the chairs for that will be Professor Niels Ben Kwashi and Dr. Ajua Padiki Nate. And just a reminder, the plenaries have a total of 20 minutes. That's 15 minutes plus 5-minute questions. And the fellow session are 15 minutes. That's 12-minute presentation, 3-minute questions.
So the next presenter, my mentor, and as well as the mentor for a lot of the people here who are into malaria. His talks have always been very stimulating. So I want you to just stay relaxed and, and listen to Professor Lasweed.
Thank you, Ben. Thank you to the organizers for inviting me to come and speak, despite I've been here several times before. And a particular thanks to, of course, to my longtime brother and rascal friend, Gordon Awadara. But I have known him since he was like this. Now I can only hope that there will be some slides at some point, because otherwise what I will tell you about actually is some recent observations that we have made, and in a way some theories also, and I will try to present the case and to see what you think about it.
Sorry, there's a little technical problem. It looks like your PowerPoint is missing for now. So, so we want to plead with you to allow the fellows to present whilst they look for your PowerPoint.
They've lost my PowerPoint.
You can't find it. It's not lost, but it can be found it now. So please, if you can just...
So I step down again?
Yes, please.
Yeah,
oh, thank you very much.
That was easy.
For your
So we would go ahead with the fellow session. Our first presenter is Christopher Abana, and he would be presenting on epigenetic modifying compounds identified as HIV latency reversing agents in vitro.
All right, good afternoon. My name is Christopher Zabian Abana. I'm sure most of you sitting here might be wondering, ever since the identification of HIV in the early 1980s, close to more than 38 years ago, you've been wondering why we wouldn't be able to find a cure. And even that, is a cure really needed? Well, we know that ever since HIV came, we've been able to. One is huge economic burden for people living with antiretroviral therapies, even in low and middle income countries like part of the world. Economic burden for getting antiretroviral therapies, even access to the ARTs. Importantly, we can also talk about HIV drug resistance. And talking about this, if a cure is needed, do we even Good. Thank you. So, even explain to you why we need a cure. Even talking about why we need a cure, why has there not been a cure for the past 30 years? We realized that COVID came in 2019, 2020, and within the shortest possible time, we've been able to get vaccines that have been able to combat this deadly COVID-19 disease. But we can't say the same thing for HIV. And even even if we're talking about cure, how does the virus? Second is of them. This gets stem cells donors who are, and then we have then we can also talk about the shock and kill. So kick it to come out from the system, and then by doing that, because if you are there, then we just find molecular mechanism of the shock and kill. Now they got a bigger megaphone by making a. So shock and kill a genetic like my point is. Okay, we can see that the DNA is a tighter, it's able to wound around it so they can get it to us. So if you want to, and then there'll be transcription. Gene, to first of all, cell group, the gene is switched epigenetic compounds in that on without going off. So there are different being transcribed, it can be cleared by the antiretroviral therapy. Example of HDAC inhibitors that have been used, that scientists, we've seen the use of volonostat, the use compounds to try to find potent latent reversing agents. Transferase, we have histone the cell model system. The envelope protein of the virus is knocked off to cause infection. Then we also have the negative cell line is latent. So then I do my flow scan. I realized that, but most of them have shown effects as latent reversing agents before. So this is the graphical representation of the 7 compounds that had effect on latency reversal. We could see that one of the compounds on HDAC, that's MC1568, that is showing like 91% GFP protein expression. We screen them in a primary T cell model, because the first model that was going to patients. So I look for, because the whole idea is to use HIV patients, to try them with HIV patients. So we'll take patient cells that they are virologically suppressed for more than 6 months, take up their resting memory T cells, and then challenge them on the using these compounds to see the effect of these compounds. And then we'll try to also measure we report the establishment of a low to reactivate HIV in primary cell latency cell system and in patient CD4 T cells ex vivo. So I'd like to thank my collaborators and supervisors, and thank you so much for paying attention.
That was very, very interesting. I have a question on the specificity of these compounds, because I assume that you are targeting an enzyme that is present in all cells. Can you hear? I assume that you target an enzyme that bounds to specific cells infected with the virus, because I assume that it would affect other things in the cells.
I didn't really get some of the things that you said.
The specificity of the compounds in relation to HIV, because they are affecting a central enzyme involved in epigenetic changes. If you have this turned on all the time, presumably that could be toxic.
Yes. So I get your question. So if you have it the gene turned on all the time, the target gene that we are talking about is only HIV. Other cells are not going to be affected. So because the human ancient days that have been living with us for so long without coming out, because it just says that these compounds do not have any effect on those live with us for some time, so there's not going to be any effect. The target is only for HIV. Yeah.
With your answer. So I think you need to check it again, because I think that's why it's called shock and kill in part also, because there is going to be a toxic effect. But the idea is that the duration of the toxicity will be shorter than the duration of treatment. And then my question is more of a, I guess, philosophical one. Whether, you know, in your intro you talked about life expectancy is still reduced. Is it really? If you're treated with the state of the best drugs? Or is it an issue of it's important to do this here because we still don't have the best drugs?
Yeah, so I mean, it's not like in the larger scale because not everybody's be able to get access to those drugs. So that is why I said that.
Just a consideration when you go into human cells, right? You may want to get some sequence information on. Does that make sense? Be important to capture the sequence of the virus, okay, because it can be highly variable.
Yes, please. Thank you.
Okay, thank you very much.
Thank you,
Christopher. Our next presenter is Edem Adika, and he would be presenting on molecular determinants of Plasmodium falciparum ex vivo antimalarial drug responses in Ghana.
Good afternoon. I'm Edem Adika, and I'll be sharing with you some work I did for my master's here on molecular determinants of Plasmodium falciparum ex vivo antimalarial drug responses in Ghana. So we've seen that malaria remains a public health problem. What I'll be talking about are drugs, even though others will make more case for vaccines. So the current effort is to eradicate and eliminate malaria, but this is threatened by various factors like vector insecticide resistance, invasive vector species, and gene deletions that are compromising diagnosis. And what I focus on is drug and drug resistance. So these interventions are used here in Ghana, ACTs, which are the frontline treatment for uncomplicated malaria, intermittent preventive treatment in pregnant women, and also seasonal chemo prevention in children. It's important to safeguard this intervention, and we are making a case for nanopore sequencing, which offers portable, cost-effective sequencing that can generate real-time data in order to monitor the effectiveness of this approach. So current data shows independent emergence of partial artemisinin resistance in Southeast Asia, even though it was expected that it would spread from in East Africa, although it was expected it would spread from Southeast Asia. Also, data from West Africa shows increasing tolerance to partner drugs, particularly lumefantrine. That is important drug pressure. So this work tried to answer the questions on what are the ex vivo responses of P. falciparum clinical isolates to frontline anti-malaria drugs in Ghana. What are the prevalences of validated molecular markers that confer drug resistance in this isolate in Ghana? So our overall aim was to determine the ex vivo responses of P. falciparum clinical isolates to frontline anti-malaria and the molecular determinants of different responses to this drug. This was achieved by determining the ex vivo responses to dihydroartemisinin, artesunate, which are ACT derivatives, to the prevalence of validated molecular markers in we performed a study at the Lekma Hospital in Accra, and we recruited participants who were positive for malaria, and we excluded those that had taken anti-malaria 2 weeks to the 6 mils of blood we used for our ex vivo drug assays within 4 hours. We stored an aliquot for because we look at the median age and the range, could see that malaria still affect wide range, even though children under 5 are the hardest hit. And also, majority of the participants, other studies that have shown that urban malaria is drug in this clinic, which is expected as this an urban population, incubated with serially diluted drug compound for 72 hours at 37 degrees Celsius, after which we stain with cyber green light with graphpad prism to generate IC50 values. And if you look at our values, so far we've performed our 48 phenotyping with good as the best being 43 out of that for chloroquine. If you look at our data here, the lower the IC50, the more effective the drug. So we could see that dihydroartemisinin and artesunate are the most effective, which is expected because these are artemisinin derivatives. However, if we look at our partner drugs, looking at lumefantrine and amodiaquine, lumefantrine has an IC50 twice that of amodiaquine, showing that amodiaquine is performing better than lumefantrine. Chloroquine, which has been withdrawn close to 2 decades in Ghana, is showing some efficacy better than that of lumefantrine, which is in variation in the distribution, with some isolates nearing or just going above the cutoff for resistance, whilst that for chloroquine, none approach that. Again, this could be attributed to low drug pressure, since this has been withdrawn and there's a lot of drug pressure from the front of these ACTs. Then we went on to compare this data to prior work done by Prof. Kwashi here at Noguchi, and we saw that there's minimal increase in the geometric mean IC50 for DHA and artesunate, which are the ACT derivatives, again showing that they are effective. And for the partner drugs, there was an increase in the geometric mean with that of lumefantrine for chloroquine, which again shows that there is gains in efficacy to chloroquine. So in summary, for our phenotyping, we've shown that the artemisinins remain potent, and for the partner drugs, lumefantrine and amodiaquine, due to increased geometric mean IC50s over there, there's possible tolerance to this drug, and also the data support other studies that have shown that there are gains made in the efficacy of chloroquine. So we went on to, sequence the 22 of the phenotype clinical isolate. So we extracted DNA, multiplex PCR was performed for 5 markers: P-falciparum, chloroquine resistant transporter, multi-drug resistance, dihydrofolate reductase, from this gel, we show that we've been able to successfully amplify all our 5 markers. That our sequencing was successful with gross coverage for our distribution of these per the 22 isolate. 50 coverage is enough to. We went on to look at these 5 markers. I'll start with the antifolate markers, and if you look at folate had triple mutations at these positions. That's a majority of our isolate were double mutant and to sulfadoxine. However, only 1 isolate carried a mutation that preventive treatment. If you look at the CR disease, and are expected to be sensitive to chloroquine, which ties in with our ex vivo phenotyping. For PFMDR1, interestingly, all our isolates carried the wild type lumefantrine, which again is showing that, well, this marker resistant allele. The phenotype based on to artemisinin, what I didn't show is that for the artemisinin local, we didn't find any Kel-13 polymorphisms that are known to confer partial resistance for intermittent preventive treatment. So this is a manuscript we submitted on part of the work which is under review. In summary, from our molecular study, we've shown that all our isolates carried a wild-type allele for CRT, and there were no Kel-13 polymorphism known to confer partial artemisinin resistance, which is expected. And also, antifolate resistant markers remain high in Ghana. However, we didn't find mutations that confer high-grade resistance to sulfadoxine pyrimethamine. Thus, we could still use